So today I'm doing a comparison of the Oxford AstraZeneca vaccine, the Pfizer vaccine, and the Moderna vaccines, which were the top 3 vaccines used during the COVID period. So this is a little summary on the Pfizer vaccine. It's also referred to as these two names, and it is an mRNA vaccine, and I'll talk more about that on the next couple of slides. The vaccine is given in 2 doses. All of those 3 vaccines were given in 2 doses. And it must be kept in ultra cold temperatures, and it only remains stable at room temperature for up to 6 hours. The vaccine also has high production costs, mainly because it is an mRNA vaccine and they cost more to produce. And it was also sold to make a profit by the company, which means that in countries that are developing, they can't really afford to buy this kind of vaccine in bulk for their population. And they don't have the expensive infrastructure that's needed to keep the vaccines at the correct temperature for the period of time that it's needed. So less of these vaccines were used globally, more so they were used in the US. Okay, so you have the Oxford AstraZeneca vaccine, which was created here in the UK by Oxford University and the company AstraZeneca. They came together to create the vaccine. It has these alternative names in scientific papers, and it's delivered using an adenovirus vector, which I'll also talk about in the coming slides. It's given in 2 doses, and the second dose is given around this time after the first dose. The key thing about this vaccine is that it only needs to be refrigerated at about 2 to 8 degrees, which is the general range of a refrigerator, which makes it a lot more accessible to those developing countries that couldn't afford the infrastructure needed for the first kind of vaccine. As well as that, the company, AstraZeneca, came up with a non-profit agreement, which meant that their vaccine could be sold a lot cheaper than the other vaccines. I think the difference was like $10, $12, $15 in between each individual vaccine. This one was sold for $2 to $3, which means that it was used a lot more globally than the other one. So how do the Pfizer and Moderna vaccines work? Both of them are mRNA vaccines. This mRNA is encapsulated in lipid nanoparticles and injected into the body. The lipid nanoparticles are what allows it to enter your body cells. The mRNA is a molecule that enters the cell and instructs it to use its own machinery to create the spike protein of the COVID-19 vaccine. And the spike protein is what allowed the virus, the COVID-19 virus, to enter the cell and replicate inside it. It's very specific, it's like the antigen. So if I draw that for you, you have your cell and you have your nucleus, and your mRNA is injected into the body, but then it travels into the cytoplasm, and then you have your ribosomes that read it, and it is and it's translated into the spike protein, which is this, and this spike protein is then presented on the outside of this cell, and the immune system will identify this cell as, and it will create an immune response, basically. The difference in how the Oxford AstraZeneca vaccine works is that it is a vector vaccine. Do you want to? It's a vector vaccine. It uses the chimpanzee adenovirus as a vector for the DNA, which contains the spike protein. So you have your, let's say, chimpanzee adenovirus, and then say this is your DNA, which is different because that one was RNA, this one's DNA. In this, the portion of the DNA that allows this virus to replicate, the adenovirus from the chimpanzee, it's deleted by the geneticists, and it's replaced with this little bit here, which is the DNA that codes for the spike protein. This is then extracted and injected into here. And it will then enter your nucleus. Very scrappy drawing. And then that is converted into RNA, which undergoes the same process here. It becomes mRNA when it leaves the nucleus, is translated by the ribosomes, and becomes the spike protein. The reason why they use chimpanzee adenovirus is because your body has a less chance of having a pre-existing immunity to the adenovirus. There is a human adenovirus, but the chimpanzee one works better, and it allows it to be used globally rather than, you'll have to stop at some point when people develop a resistance. Yeah. So, comparing the efficacy of these vaccines. As you can see on the board, these vaccines seem to have a much higher efficacy than the Oxford AstraZeneca vaccine. And whilst this is one way of comparing how well the vaccines work, this isn't really the only way to look at them. So what is efficacy and how is it decided? So there's a trial, and the trial is split into a placebo group and a vaccinated group. Of those that end up with an infection at the end of the trial, the number of individuals in the placebo group and in the vaccinated group determine what the efficacy is. So if you have 100 people, like it says there, and 50 of them are in placebo and 50 of them are vaccinated, then it means that the vaccine didn't really work. It didn't have an effect. The same amount of people would get the would get the virus whether or not they were vaccinated. If of the 100 people infected, 85 of them were in the placebo group and 15 were in the vaccinated group, then the vaccine has an efficacy of 85%. Having the vaccine means that you're better protected against the infection. This statistic means that each vaccinated person is 85% less likely than a person without the vaccine to contract an infection every time that they're exposed. It doesn't mean that of the population that is vaccinated, only 85% are protected, which is a common misconception. So why can't efficacy be the only comparative point between vaccines? Each COVID vaccine's efficacy is calculated in the same way, but there are different conditions that each trial went under because it was, very much at that time, vaccines were needed immediately, so the conditions weren't kept the same for each of the vaccine trials. These are two key points that affect the efficacy in some sort of way. So the rate of transmission, so the amount of COVID cases at the time that you were conducting the trial, means that if you had more COVID cases during the trial, then more people are going to be in the placebo or the vaccinated group, and that will affect the efficacy. But as well as that, the country that the trial takes place in, that also affects it, mostly because there are different strains of COVID in different countries, but as well as that, different countries also have different case rates, so you'll have more in a different country, and then that one won't be as effective with the vaccine, and so that changes the efficacy as well. So you have a comparison of these trial conditions. This is for the AstraZeneca vaccine, and then I've put Pfizer and Moderna together because their trial conditions were relatively similar. They happened at around the same time. So the trials occurred for the AstraZeneca vaccine when there were variant strains circulating. It happened later on than these ones. And this one mostly occurred when the original strain was predominant, and mostly in the USA, so there weren't that many different strains of the COVID vaccine at the time that the trial occurred. And you can see here from these statistics that of the 43,000 in the Pfizer trial, 38,000 were from the US, so it was very limited to the US population and the US strains. When it comes to the AstraZeneca vaccine, the trials predominantly occurred in UK, Brazil, South Africa, and there were lots of different kinds of strains in these different countries. For example, the vaccine wasn't very effective in South Africa because that strain was a lot different to the one in the UK. The trial was the largest COVID vaccine trial of its time, so the AstraZeneca one, and these statistics show that it was there was a pretty good range of where each population was from. So we can talk a bit more about AstraZeneca's interesting trial results because they did their trial a little differently. They had a bit of an accident when they did their trial. So you have your people in Brazil and you have your people in the UK. About 9,000 people in Brazil were part of the trial population, and about, I think, 3,000 were in the UK trial population. The way that they did it is that there were two doses, so the first dose and the second dose. And in Brazil, they the correct way. So they had their first dose, which was the entire dose, and their second dose, which was also the entire dose. But by some mistake, in the UK, they managed to give the first dose as half of the dose and the second dose as the entire dose. And this drastically changed the efficacy of the vaccines. So the Brazil vaccine efficacy was 67%, but for the UK, it was 90%. So this difference in how the vaccines were administered changed the efficacy hugely, which is really interesting to look at. Get rid of that. So there was also some controversy surrounding the AstraZeneca vaccine. Mostly the news focused on the USA, which has to this day banned the use of the AstraZeneca vaccine in the US, even in emergency use, which some European countries also banned the AstraZeneca vaccine in their countries but opened it back up for emergency use. And this was for two reasons. This was because in March 2021, AstraZeneca announced that their vaccine had an efficacy of 79%. But then a lot of countries, they looked into this data and they realized that there were some discrepancies, and AstraZeneca had to come back out and say that their previous data was wrong in some senses and it was reduced to 76%. And they lost a lot of trust with other countries' governments over this. But as well as that, there were huge concerns surrounding a rare side effect called thrombosis, which causes blood clots. And a lot of countries immediately after finding this out stopped the import of the AstraZeneca vaccine into their countries. But it was later found that only 1 in 100,000 people were at a risk of developing it, and they had to have some previous factors that led to it, so it wasn't really a huge concern. So this diagram shows outcomes of exposure. So you can get the COVID infection, and it can cause this spectrum of things. You can have no infection at all. You can have no symptoms. You can have moderate symptoms, severe symptoms. You can be hospitalized, and you can die. The vaccines can target different areas of the spectrum, and the AstraZeneca vaccine aimed to eliminate infection entirely. So they said, we don't want anyone to get infected in the first place, so they would consistently test people throughout their trial periods, and anyone that had the virus, whether they showed symptoms of it or not, they were noted down and put into that category. But with the Moderna and Pfizer vaccines, they focus more on preventing the symptoms of COVID, which is also extremely valid because you can be infected with COVID. It was likely that you would be infected with COVID. What they were trying to do was prevent people from developing harmful symptoms that would result in them being hospitalized or, dying from it. And I think the key thing here to notice is that not a single vaccinated person in any of the trials, in any of the top, I think, 5 or 6 vaccine trials, were hospitalized or died from COVID. So it's a lot better to get vaccinated. It doesn't matter which vaccine you get. It was a lot better to get vaccinated than not get vaccinated. Thank you for listening.